It has killed more people than all the wars in history combined. It is stealthy, ever-changing, elusive, and inevitable. It knows no boundaries, no age, race, or region. Cancer is responsible for over 500,000 deaths each year in the U.S. Worldwide, the number is over 7 million. Yet there is a war being fought against cancer, carried out by people who, for the most part, toil in quiet purpose. One such person is George Vanderwood. In the war on cancer, Vanderwood has risen from soldier to general, producing critical discoveries in his lab, but perhaps more importantly, inspiring generations of brilliant minds to join him in his crusade. In January of 1971, U.S. President Richard M. Nixon turned his attention from the failing war in Southeast Asia to a war that hit closer to home, and one that seemed equally unwinnable.
I will also ask for an appropriation of an extra $100 million to launch an intensive campaign to find the cure for cancer, and I will ask later for whatever additional funds can effectively be used. The time has come in America when the same kind of concentrated effort that split the atom and took man to the moon should be turned toward conquering this dread disease.
It was a bold declaration, and a generation of young scientists answered the call, one of whom would play a critical role in shaping the institution that ultimately would lead the effort in taking the fight forward. Born on Christmas Day in 1935, George Vanderwood was raised in a modest, middle-class neighborhood in Queens, New York. George's parents gave him a loving, nurturing home, a strong work ethic, and a well-developed sense of humor. These attributes, along with a tremendous curiosity, would from time to time get George in a little trouble. They would also develop into the qualities that would serve him well as a scientist and as a leader.
George probably played the most tricks on me.
I had the chemistry set. I set the house on fire once with a special concoction. So
There were some chemist things he was mixing, and if you stepped on them, sparks would fly. And there was a guy who delivered soda one day, and I guess these sparks flew, and he dropped a case of soda. I think George was inside laughing at the time.
no, it wasn't a canary.
It was
a
parakeet? Or
parakeet.
It was a parakeet.
Mom had a parakeet that died, and I wanted to see what it died from. That's all. It was such a big deal. So
I didn't remember it being dead.
It was dead.
I just remember growing up, I thought it was pretty unique that we really had a loving household and a lot of fun.
Well, I was glad I got. Let me have you one on a certain day, and that was Christmas Day, and that was really American to have you. Only thing that I shouldn't have named you George. I should have named you Jesus Christ.
After serving in the U.S. Army, George completed his undergraduate work at Hofstra College. In 1959, George married his neighborhood sweetheart, Dot Staple. George selected Rutgers for his postgraduate work. While at Rutgers, he was mentored by a man who would have a tremendous impact on how he would manage his own lab and inspire others around him.
I was very lucky to find Frank Davis and to choose him as a mentor for doing my graduate studies, learning the process of how to be productive in science, how to be creative, how to best be creative, free thinking, and he provided those opportunities for me, and those are opportunities that I try and mimic.
George, now armed with a strong education and his own concept of effective leadership, made a critical decision, one which would later position him to accept the challenge of a lifetime. The decision would take George to a somewhat remote outpost and the firewall of highly infectious disease in animals, Plum Island.
Every morning, commuters board a ferry for work. Not an unusual occurrence in this country, but these commuters are unusual. They're employed by the United States Department of Agriculture. They're going to man what is perhaps the most unique laboratory of its kind in the world. Different people, different skills, but all working toward one end, to foreign animal diseases out of the United States. A small island off the eastern tip of Long Island, New York, was selected. Plum Island, a former coast artillery post, is now the U.S. bastion against foreign animal diseases.
In 1954, they designated Plum Island to be the place where the Department of Agriculture could have a research station and a diagnostic center. The diagnostics were to provide tests for a number of exotic animal diseases.
When it authorized establishment of the center, Congress specified that it be located on a coastal island. Congress also required complete federal jurisdiction over the island, so that movement to and from could be strictly controlled.
It was chosen because of its isolation, because of the high contagious aspect of foot and mouth disease, highly infectious virus. It was a great opportunity for young people to come and train in animal virology. One of the reasons I came was it was the only place where they were making high concentrations of an animal virus, which allowed you to study it much more easily, find neutralizing antigens, understand the virus when it infects cells, all of those things.
But he was into everything, he's so interested so wide, and he was an excellent person to have to call on for questions about different things. George always knew or could help us find out. So he was one of the more popular people that worked on Plum Island, I think, over the years.
George spent several years building his credentials as a leading scientific researcher on Plum Island. His experiences using the high containment facilities on Plum Island and recombinant DNA at the National Institutes of Health, or NIH, enabled George's lab to clone the first oncogene, a tremendous stride forward in the war on cancer. In addition to building a reputation for excellence in science, George began to emerge as a quiet but effective mentor and leader. This combination of characteristics would serve him well in years to come when he was appointed by National Cancer Institute Director Vince DeVita to lead the Advanced Biosciences Laboratory's basic research program at the NCI's Frederick, Maryland facility. It was in this role that George truly made his mark, not just for his strides forward in science, but for taking the reins of an organization in chaos.
The institution of Fort Detrick has a very checkered history. It began as a center of excellence for biological warfare during the Cold War, when anthrax and smallpox and deadly pathogens were being grown by the Department of Defense. In a show of amazing acuity, Richard Nixon showed up at the front gate of Fort Detrick in 1969, said, "I'm going to stop the biological warfare of Fort Detrick and pretty much turn this place over to the National Institutes of Health."
While the location of the National Cancer Institute had dubious beginnings, it was the problems inherent in the structure and makeup of the organization that would prove to be the greatest challenge to DeVita and George Vanderwood.
When I became director, Frederick was a garbage pail. We had a good hard look at it and decided that we needed to make it first class. And so we eventually offered George the opportunity to take it over and build it. And he built one of the finest basic research facilities in the country.
At the time we began, there was stovepipes of knowledge. People who were trained in chemical carcinogenesis couldn't talk well with people who were doing cell biology, couldn't talk well with people doing virology. Different buzzwords, different language, different acronyms. And it stayed that way for a long time because no one really understood what the basis was.
Traditional research that was going on in those days was to take a mouse, fill it full of a bunch of chemicals off the shelf and see if it got cancer on one side of the hall, and then on the other side of the hall are a bunch of guys who took the animals with cancer and then took the same chemicals and stuck them in and asked whether or not the tumors went away. I mean, it was really just trial and error.
George quietly began to make the changes necessary to eliminate the professional silos and break down the walls between researchers that inhibited new ideas and leaps forward in discovery. In the process, George's own lab began to publish discoveries that would significantly move the war on cancer forward.
When I think of George's contributions to the field, I think of his work on metastases, which certainly turns out to be critical or very important in terms of how tumor progresses from a more benign state to a metastatic state.
My lab discovered a molecule called MET, which has a function that for transmitting signals from the outside environment of a cell to the inside to tell the cell how to deal with its environment.
This protein MET, the receptor for hepatocyte growth factor or scatter factor, he knew that it was associated with tumor growth. And that would have been enough for most people, but he also was perceptive that it changed the motility and it changed the appearance.
A very important characteristic of tumor cells is when they shift from growing and proliferating to becoming invasive and migrating to distant sites, where again they can have to acquire characteristics to grow as tumor cells.
The work that my dad did, and along with other people who were looking at the mechanisms of what causes a cell to become a cancer and what allows it to go around the body and make metastasis, are dependent upon very small molecules that are normal proteins in the body. And by figuring out on a more precise level when those particular proteins are malfunctioning, and then making a drug that's very specific to inhibit that particular protein, it allows you to have a very small target for the cancer itself.
He developed this concept of mitogenesis, motogenesis, and morphogenesis. Not only does it grow, but it moves in a different direction, and it changes this shape. Most people would focus on one of them, but he focuses on all three of them at one time. And although not a physician and not trained in pharmacology and not experienced in the intricacies of the pharmaceutical business, George has decided to bring this into the clinic. And so he's made pharmaceutical people believers.
It's almost like a designer medicine, and there have been multiple drugs that have come out in the last 5 or 10 years that allow targeting of very specifically these malfunctioning proteins. A lot of these are natural products or molecules that have been around for a long time, but now that we know the mechanisms that they inhibit, they can be used in different ways to try to reduce cancers.
The contributions he's made in areas of viral oncogenes and in the area of the MET receptor and HGF have absolutely direct impact on human health. And I think that's the kind of legacy that all of us want to leave behind, a very successful history of discovery that ultimately will lead to better human health.
While George's discoveries began to ripple out into the scientific community, his leadership style began to transform the National Cancer Institute. This style, informal yet intense, cooperative yet competitive, began to take root and inspire his colleagues and the young researchers hungry to make their own mark in the war.
His lab was a unique group of people who interacted extremely well. Part of that was they could all tolerate this somewhat unusual approach to science that George had.
George had that unusual characteristic that he was brilliant and nice.
George finds the best people that he can for different positions and then let them do what they think is best and intervenes only to the extent that he sees it being necessary.
His ability to convey to younger people the excitement that he has and the passion that he feels for the work that he's doing.
He was happiest when he saw people that he brought in sort of grow and become really the kind of scientists that he thought they could be. The most dangerous thing you could do was to walk into George's office and tell him an exciting new result, because he would immediately take you in this bear hug. It didn't matter if you were male, female, he would take you and crush you. And so you tended to be, you had to sort of be prepared for this, or else you might end up with a fractured rib.
George's hugs become a reward for those of us who work with him.
But this was his way. He was excited about everything, that all the science that he was doing, all the science you were doing.
With the introduction of the personal computer and other research tools, methods evolved rapidly, and the volume of vital data mushroomed. Communication and cooperation increasingly became a critical weapon in the war on cancer. In 1985, George and his colleague from the Salk Institute, Tony Hunter, founded the Oncogene Symposium, creating a new and brilliant forum for ideas to fight cancer. By bringing together the best and brightest to collectively explore the root causes of cancer, George helped foster the next generation of cancer researchers, while breaking down the national and international barriers to cooperation that stood in the way of significant advancement.
The Oncogene meeting, which he started and ran for 20 years. This meeting gathered together young scientists, senior scientists, graduate students to full professors. Everybody was equal at this meeting, and we were all discussing our results.
The best ideas emerge when a wide variety of ideas are openly discussed. And I think it's one of George's gifts to attract people with strong opinions and to really attract people with divergent opinions, and yet bring them together in a way that allowed them to interact successfully.
The greatest thing was it provided an opportunity for these young people to meet colleagues who they would share the rest of their career with.
With the tremendous strides forward in human genome sequencing in the 1990s, the potential impact of personalized medicine began to emerge. Customized protocols based on an individual's DNA showed tremendous promise in efforts to fight cancer more effectively. George, deeply driven to move the discoveries in the lab more quickly to the patients suffering from cancer, recognized the importance of translational medicine and made it the focal point of his efforts in his personal war on cancer.
Translational research can be many different aspects of taking basic research and turning it into something that can help in a clinical setting. So it can be development of novel drugs for treating different types of cancers. It can be the development of new types of diagnostic tools that help us tell whether someone has cancer or what types of treatments are most suited for that particular individual.
It was a great achievement marrying all of these separate disciplines, and it continues today. The process that we are trying to do is marry all of what we have discovered about the process of cancer to the medical community.
Even as translational medicine has become the watchword in moving both research and treatment forward, the burden of bureaucratic regulation threatens to hinder the efforts of the scientists and physicians determined to move forward. Getting potential therapies to suffering patients takes millions of dollars and many years. And in the case of terminal patients who could immediately benefit from new protocols and experimental drugs, the system to sanction experimental treatments has become unwieldy and obstructive. In many cases, preventing those dying from cancer the chance for more time, or perhaps even a cure.
We've actually won a good bit of the war on cancer. What we haven't won is the war on the cancer establishment. You compare what we know now compared to what we knew when the Cancer Act was passed, just like night and day. We have a critical mass of usable knowledge. We know enough about the pathways that a normal cell, that are aberrant to a normal cell to become a cancer cell, and how a normal cell becomes a cancer cell, to take a shot at controlling these. The problem that's standing in our way is we no longer have the freedom to do that.
There's no question that the bureaucratic and administrative burden put on scientists impede the search for new discoveries. We have developed too many layers of bureaucracy. There are too many difficulties that I think are very troubling to scientists.
You know, if the FDA regulates us to the bone, we can't get it by a Human Investigations Committee. We don't have enough coordination across pharmaceutical companies, which needs to be done at a national level. So we've lost the war on, the flexibility we need to use the knowledge.
While the war on cancer began before Nixon, the declaration of war on cancer and the resulting efforts have had a huge impact. The rates at which people are developing and dying from cancer are in decline for the first time in U.S. history. In coming years, these strides forward promise to be even more significant. And while for some the war is over, George Vandewood continues to fight. His legacy includes serving for a decade as founding research director of the Van Andel Institute in Grand Rapids, Michigan. George helped create a completely new institution incorporating both basic and translational research, where ongoing investigations into the genetic, cellular, and molecular origins of cancer promise to have an impact on finding cures. And though he will hand over the reins of leadership at the Institute, George continues to use his lab to bring the war to the enemy.
I didn't know he was going to go on the big things he's gotten into today, He's a good scientist, and of course George knows how to get work out of people, and he knows how to put the priorities where they belong. And in hindsight, I'm not surprised he worked up where he did, because he's a kind of a man you enjoy working with.
He has a way of making everyone believe that the enterprise he's connected with is going upwards. It's going to succeed. It's going to be better. It's going to be stronger. We're going to get the best people. We're going to do a good job.
He was able to raise the level of science here to such a high degree that it's having a lasting impact on all of us who ever work with him and continue to work here, independent of him, but still working under his legacy of scientific excellence as being the #1 goal for all of us.
I think there are going to be a lot of people who are going to trace their lineage back to George, and that's going to be, that's his biggest contribution.
There are very few people who can say this. Very few people who have a combined legacy of outstanding science and really special, novel leadership style that develops other leaders and develops young talent.
George was a general, for sure. But he was Patton. Okay. I would say I was Eisenhower. He was George Patton.
He cared a lot about excellence, and I think that if In Search of Excellence was written in 2008, that probably there would have been a chapter on George.
Discovering something, helping to bring it forward, seeing people treated with a drug, a result of that, that's enough. I'm not greedy.